Hello guys, welcome to my channel. So as you know, we just finished in the last video the entire cell cycle. So the next topic today we'll discuss about the cell numbers and the cell size. Cell numbers and cell size. Right? So like if you know, a fertilized mouse egg and a fertilized human egg are similar in size, about 100 micrometer in diameter. Yet an adult mouse is much smaller, of course, than an adult human. So what are the differences between the control of a cell behavior in humans and mice that generates such a big difference in size? Right? So that question arises, what are the difference? So there are three fundamental processes largely determine organ and body size. There are three fundamental processes, right? So the number one is cell growth, right? Number two is cell division, and number three is cell death. So these are the three fundamental processes that largely determine the cell growth, right? That how the organ and the body size will become. So these are the three fundamental, and today we'll discuss this, right? And each of this process in turn depends on a program intrinsic to the individual cell, regulated by signals from other cells in the body. So these are actually in turn related to some other signals from the other cells of the body. Right, so first we'll see how the organism eliminate unwanted cell, how the unwanted cell are removed from the body. So the as we know, the cells of a multicellular organism are member of highly organized community, right? So like such as, so we are a multicellular organism, and this entire body is actually a highly organized community. It is organized at every single level. So the number of cell in this community is tightly regulated, not simply by the controlling, it's just like not simply by the controlling the rate of cell division, but also controlling by the rate of cell death. So if the cells are no longer needed, they can commit suicide by activating an intracellular death program, a process called programmed cell death. Programmed cell death, and that is called apoptosis. Right, so this is the process. If the cell are no longer wanted in the community, they can commit suicide by activating this intracellular death program. Right, so in animal cell, most common form of program cell that is called apoptosis. Right, this is the most common program cell that. So the amount of apoptosis that occurs in both developing and an adult animal tissue can be astonishing, right? Why? Because in the developing vertebrate nervous system, for example, more than half of sometimes of nerve cell normally die soon after they are formed, right? And in healthy adult humans, billions of cells die in the bone marrow and intestine every hour. So that seems very remarkably wasteful, right? For so many cells to die, especially as the vast majority of the cells are very perfect and healthy at the time they kill themselves. So, like the question arises, what is the purpose for this massive cell suicide? Right? So, like in some cases, why? The answer is very clear. Like for example, mouse paws on our own hands as well and feet are sculpted by apoptosis during the embryonic development. Right, so when we are in the embryo, so what happens? So like when our hands are sculpted, so actually it's shaped something like this. So this is something like spade-like structure before, and the individual fingers and the toes. So before the fingers and toes looks like this, and then the individual fingers and toe, the cell starts to die, and then it becomes over like this. We have hands and toe, right? So this is the reason why the cells are dying, right? In other cases, cell die when the structure no longer need them. So for example, it forms already a structure, they form and they do no longer need the cells, so then the cell will die. Like for example, when a tadpole changes into a frog at metamorphosis, the cell in the tail die, right? Because as you know that a tadpole has a tail but the frog does not have a tail, so like when it turns into the frog, so the tail part, all the cells died and die in that part, and then it becomes frog and the tail falls off. This is one of the reason, right, why the cells are removed by apoptosis. So in adult tissues, cell death usually occurs exactly balances cell division unless the tissue is growing or shrinking. So for example, if a part of our liver is removed in an adult human or adult rat, so the liver cell proliferated to make up the loss. So then the cell will divide. But if, for example, if our liver is enlarged, right, by any medication or anything, so what will liver do? Liver will greatly increase the cell death and try to be in the normal size until the organ has returned its original size, right? So these are some of the reason why the apoptosis happen, right? So the apoptosis is actually mediated by an intracellular proteolytic cascade. So what does that mean? So cells that die as a result of acute injury typically swell and burst, spilling their contents all over their neighbors. So like what happened? So when there is an acute injury in any kind of like that to a cell damage and the cell has to die. So like the cell dies and like whatever contents it has, it spills all around its neighbor. So that means so there will be an acute inflammatory response, and that is a very damaging inflammatory response to heal that thing, right? And so what happens, so the neighboring cell also are under the damaging inflammatory response, and that will result in a cell necrosis, and that is a process called cell necrosis, right? So cells that die as a result of acute injury typically swell and burst, spilling their contents all over their neighbors. That is a process called cell necrosis, right? But by contrast, when the apoptosis happen, it does not trigger any inflammatory response. It dies without damaging its neighbor. How? So the apoptosis, in apoptosis, the cell first bulges and then it shrinks and condenses, and the cytoskeleton of the cell collapses. The nuclear envelope disassembles and the nuclear DNA breaks up into fragments. But what all this happens in within the phagocytic cell. So most importantly, the cell surface is altered. So the outside of the cell is altered in such a manner that it immediately attracts the phagocytic cell, that is the specially killer cell, usually phagocytic cell that are called macrophages, right? Write it down, phagocytic cytic cells, especially macrophages. So this cells is responsible, so they will engulf the apoptotic cells, whichever cell is going to for the apoptosis before it spill their contents all over, right? So this rapid removal of dying cell avoids the damaging consequences of the cell necrosis, and it also allows the organic components of the apoptotic cell to be recycled by the cell that ingest it. So it's like the same like when you're throwing garbage, right? And if you're throwing garbage like just outside your house, right? So your neighbors and everywhere in your community, it will be like smelling after some days. But like, so we have a pickup every now and then, right? And we have a recycle pickup as well. So that means our community and our neighborhood will remain clear. It's the same for these cells as well. All right, so we'll see the mechanism behind this apoptosis. So what is happening for the mechanism. So the molecular machinery responsible for apoptosis, which seems to be similar in the most animal cells, involves a family of proteases that is called caspases. Right? So, all right. So responsible protease, I will write it down here. That is caspases. So this is a protease that is responsible for the apoptosis, right? But this enzyme are made as an inactive precursor. So these are not, so these are active, but like this is not directly made. So this enzymes are made as an inactive precursors that are called procaspases. Alright, so I will explain to you what it does mean. Alright, so I will show you what this means. So for example, I'll show you the picture. Let's see. Oops. Oh, this is NH2 and this is OH. So this procaspases actually activates the caspase. So each suicide protease, that is our caspase, is made as an inactive precursor, right? Precursor, that is our procaspases, which in turn, so this precursor is actually activated by proteolysis. So this is activated by proteolysis. This is activated by proteolysis. So that means that proteolytic cleavage, like splitting by another member of the same protease family. So this is split by the same protease family member. So what happened? This is our procaspase and it is cleaved around here. Around like this. So this is our, I'll write it down, cleavage side, maybe like this here. Our cleavage side. Alright, so the two cleaved fragments. So for to make one caspase, so there should be two cleaved. So for example, this is splitted. The one procaspase and the another procaspase is splitted, right? And that will make one caspase. That's right. So this is after our cleavage and assembly, then it will assembly, and the prodomains are discarded. So the prodomain are discarded, and this is our active caspase, and this is, these two are procaspases, right? So this both procaspases are cleaved by a protease enzyme, right? And both together are joined to make a one active caspase. So that is responsible for apoptosis. And the prodomain. So what does the prodomain mean? So in proteins, like this is an N terminal and this is our carbon terminal. That's why carboxylic acid and amino acid is like, that's why I made like this. So what do you mean by this prodomain? I will explain to you. So in protein, there are like pre and pro domains, usually. So when there is a cleaving of the, like splitting of the proteins is involved, so the prefix that is used as a pre-protein, like pre-protein, that is used as a pre-protein, so that means the N terminal, so this terminal from the N terminal, amino terminal, is cleaved during the secretion, right? That is mean the pre-protein. When it says pro-protein, so in that the N terminal is cleaved to make the protein active, so that why it is discarding the pro-domain. So that means this is inactive, and it, if it has to make active, this should be, it is cleaved like pro protein. So the N terminal is cleaved during the secretion and then cleaved again to make the protein active. So that means that is pre-pro protein. So this caspase is a very good example for this pro domain, right? So it is synthesized as an inactive pro protein, right? This is pro caspase is a pro protein, and that is inactive. So in this case, the pro domain is used because the N terminal region is folded into a discrete. So this N terminal region is actually folded into a structural unit with a specific function, right? So that is the definition of a domain, right? So as we studied in protein, like the proteins has a different domain. So usually the pro domain mediates interaction with other protein in a complex, right? So proteolytic cleavage then removes the pro domain, activate the caspases, and triggers the, right, the mediated response, right? So this is our caspase, so that becomes active, right? And after the caspase becomes active, so each activated initiator caspase, once this caspase is active, so what will happen, it will do a chain reaction. So this caspase will start cleavaging another procaspases as well to active more caspase. And so for example, this is active and this is known as our initiator, sorry, initiator caspase, and this initiator caspase will active different other procaspases that will active more caspases, and that caspases, for example, just make, oops, not together. All right, they activate more caspases, and this caspases are known as executioner caspases, right? These are known as executioner caspases, for example, Y, and this will in turn activate more. I will just draw like this, but like this will activate more, and the chain reaction happens, right? And some of these executioner caspases then activate additional executioner like this, and keep, and this will kick off an amplifying proteolytic cascade, right? Other disassemble and others, so this will try attack the cell that has been going for the apoptosis, right? For example, one executioner caspase targets the lamina protein that forms the nuclear lamina underlying the nuclear envelope, which allows the nucleus to enter. So it will break the nuclear lamina, and that will allow the nucleus to break the DNA, and in this way the cell disassembles itself quickly and clearly, and its corpse is rapidly taken up and digested by another cell, right? So the activation of apoptotic program, like entry into the new stage of the cell cycle, is usually triggered in all or none fashion. So, and the caspase cascade is not only destructive and self-amplifying but also irreversible. So once a cell reaches a critical point along the path to destruction, it cannot turn back. So thus it is really important that the decision to die should be tightly regulated or tightly controlled. Right, so how this is really controlled, the entire apoptosis process. So the all nucleated animal cell contains seeds of their own destruction, right? So in every cell, inactive procaspases. Inactive procaspases lie waiting for a signal to destroy the cell. So it is therefore not surprising that caspase activity is tightly regulated inside the cell to ensure that the death program is held in check until it is needed. For example, to eliminate cells that are surplus, mislocated, or damaged, right? That only cells are needed to go for an apoptotic or apoptosis, right? So the main protein that regulate the activation of caspases are a member of BCL2 protein. So the main protein that is responsible is BCL2 family, right? So this is the main protein family that regulates the activity of the caspases, right? So some member of this protein family, so the same family, some member of this protein family promotes caspase activation, right? This and cell death, whereas the same member of the BCL2 family proteins are, are also the inhibitory protein, so that can stop or that cannot promote the cell death. So two most important death proteins from this family are BAX and so BCL2 family protein, and two members responsible for apoptosis are BAX and right hand. I think symbol is fine. BAX and BAK. Right. So these two protein are responsible for the activation of the cascades. So this protein are activated, which are activated in response to like a DNA damage or any other insults, promote cell death by inducing the release of an electron transport protein, cytochrome C, from mitochondria into the cytosol. So for example, like this is mitochondria, for example, yes, not a cell but a mitochondria. I will write it down here. Mitochondria. It will have for this our cytochrome, the dotted are cytochrome C, an electron transport protein, right? So when this BAX and BAK, that is our death program members of the BCL2 family proteins, start accumulating when they are activated by any apoptotic signals, right, or stimulus. So this protein start aggregating on the outer membrane of the mitochondria. So for example, like this mitochondria receives a signal for apoptosis, and so this BAX and BAK, what it does, it start accumulating. For example, this box will say activated BAX and BAK, right? So this start aggregating in the outer mitochondrial membrane, and this, as this start accumulating, so this will leads to releasing of this cytochrome C in, in the cytosol, and that mechanism is unknown, but how they release is, but this start going outside. For example, this maybe I will just draw here something like this, right? So this is our cytochrome C in the cytosol, right? So this comes into the cytosol space, and it also has another proteins as well, right? And this cytochrome C then binds to an adapter protein. So this protein, cytochrome C, actually binds to an adapter protein. So, for example, let me just So this is for example our cytochrome. Right, so this cytochrome C then binds to an adapter protein. For example, here is our adapter protein. I will name it here adapter protein. So this cytochrome C goes and binds to an adapter protein. For example, this is our adapter and it binds to an adapter protein. So I'll write it down. Cytochrome C binds to the adapter protein, and that adapter protein then assembly. So that adapter protein then assemble into a 7-armed protein complex. So this then assembles into a 7-armed protein complex. So something I will try to make 7-arm. 1, 2, 3, 4, 5, 6, and 7. So something like this 7-arm protein complex, and it will all have our cytochromes. Cytochrome C. Right, so this cytochrome C binds to the adapter protein, and when it's bind to the adapter protein, it assembles into a 7-armed complex protein, and this complex then recruits 7 molecules, and this complex then recruits 7 molecules of a specific initiator procaspases. So this, I will write it down. This complex recruits this complex recruits specific initiator procaspases. Right, so this complex recruit initiator procaspases, and specifically the procaspase 9. So they recruit this procaspase 9 to form a structure, and when they recruit this procaspase 9, so it will form a structure called apoptosome. So now I'll just make a long one and it's right here. A recruitment, for example, and that recruitment is our, for example, this is our procaspase 9, right? So this is our procaspase 9 recruitment, and for example, this is when it is recruited. Let me make another. Seven, right? For example, this does not have, and we have something in middle, right? Just put cytochrome. See, right? So this will recruit the procaspase 9 to form a structure that is called apoptosome, right? And the procaspase 9 protein become activated within the apoptosome. So this procaspase 9 protein will become activated in the apoptosome and then go on and activate the executioner over caspases and the cascade, leading to a caspase cascade and apoptosis. So this is the entire process of apoptosis, how it happens, right? So this BCL2 family member of BAX and BAK protein are responsible for the apoptosis, right? They get the signal for the apoptosis. They will start aggregating here on the mitochondrial outer membrane, then it will release the cytochrome C with the other proteins. The cytochrome C will bind to the adapter protein and the cytochrome C will assemble into a complex protein, right, that will recruit a initiator procaspase, especially procaspase 9, and that recruitment, the procaspase and this complex will together become apoptosome, and then our executioner caspases. Right, so this is one type of a signal that is received by BX and BAK. But there are extracellular signals can also induce apoptosis. So sometimes the signals to commit suicide is not generated internally, but instead comes from a neighboring cell. So some of this extracellular signal activate the cell death program by affecting the activity of members of this BCL2 family of the protein, right? So others stimulate apoptosis more directly. So this stimulate BCL2 family, but there are other protein as well, or others stimulate apoptosis more directly by activating a set of cell surface receptor protein. So on the cell surface there is a receptor protein. Okay. So for example, this is our cell for so and it has like this. So this is our cell receptor protein, for example, and that is known as a death receptor. So that receptor. So one particularly well understood death receptor is called FAS receptor. Where should I write here? That receptor one well understood. So this death receptor is very well understood, and that is called FAS. That is a fast receptor and is present on a surface of variety of mammalian cell type. And to activate this, FAS is activated by a membrane bound protein called a FAS ligand. So this is our, I will write it down, FAS, and this is cell surface, and of course it has an adapter protein and a procaspase inside. Okay, we'll name them adapter protein, and this is procaspase inside the cell, right? And this is our FAS protein. So in order to activate this, the FAS is activated by a specialized immune cell that are called killer lymphocytes. So this FAS is activated by specialized immune cell that is called killer lymphocytes. Why killer lymphocytes? This killer cell have an ligand that is an FAS ligand, so that will activate this. So for example, this is a killer lymphocyte, and for example it has maybe like this structure. So for example, this is FAS ligand. So this FAS ligand bind to the FAS receptor and then it will activate this. So for example, this and okay, this goes to here, right? And our this is our FAS ligand, and here we have our FAS, right? FAS ligand activating FAS receptor, and once they are attached, so what happened is, so this we have our adapter protein, we have our procaspase, right? So this killer cell help regulate the immune response by inducing apoptosis in other immune cells that are unwanted, right? So by activating this FAS, so the binding of FAS ligand to its receptor trigger the assembly of the that inducing signal. So once this ligand activates this FAS receptor, so that will triggers the assembly of a collection of that inducing signaling complex. So that will induce a that inducing signaling complex, which includes a specific initiator procaspase. So here the procaspase was 9, and in this, for example, the procaspase is 8 or 10, or I will write 8 to 10, 8 or 10. So it might be 8 procaspase or it might be 10. So the procaspase, so this will happen, the procaspase will cleave and activate one another, and the resulting active caspases then active the executioner caspases, and then proteolytic cascade and apoptosis, right? So this is just a simple method, right? FAS, it has, one cell has an FAS receptor, or mammalian cell. The killer lymphocyte will have an FAS ligand. When both will join, it will trigger assembly of that inducing signaling complexes, especially in this the procaspase 8 or 10, that are cleaved, and then this response will happen. All right, so this was some of the mechanism for the apoptosis, right? So in a multicellular organism, the fate of individual cell is actually controlled by the signals from another cell. So for either tissue growth or cell replacement, cell must grow before they divide. Nutrients are not enough for our animal cells to survive, grow or divide. It must also receive chemical signal from other cells, usually its neighbor, to survive. Such controls ensure that a cell survives only when it is needed and divides only when another cell is required, either to allow tissue growth or to replace it. Right? So there are actually three major categories of that. So the survival factor that is responsible. All right, so we'll write it down here. So three factors or three categories. So one is survival factors, right? So the survival factor is that animal cells need signal from another cells to survive. If deprived of such survival factors, cell activate a caspase, right, and the apoptosis happens. So this requirement for signals from other cells helps ensure that the cell survive only when they are needed. So many type of nerve cell, for example, are produced in excess in the developing nervous system, and then compete for the limited amount of survival factors that are secreted by the target cell. So they contact. So whichever cell that contact target cell, they are saved, but the others are going to the apoptosis, right? This is the way that the survival factor work. The second is mitogens. This we studied in the cell phase, if you remember the mitogens, right? So most mitogens are secreted signal proteins that bind to the cell surface receptors. When activated by mitogen binding, this receptor initiate various intracellular signaling pathway that we discussed, if you remember, in the cell cycle, that stimulate cell division. So this cell division by promoting entry into the S phase. So this is the mitogen. So most mitogen have been identified and characterized by their effects on the cells in culture. So one of the first mitogen identified in this way is the platelet derived growth factor. So that is PDGF. So that is our platelet derived growth factor, right? So when blood clots form in a wound, for example, right, blood platelets incorporate into clots and are stimulated to release the platelet derived growth factor. So this PDGF then binds to the receptor tyrosine kinases in surviving cell at the wound site, stimulating the cell to proliferate and help to heal the wound, right? So similarly, if a part of our liver is lost through surgery or acute injury, a mitogen called hepatocyte growth factor is stimulate the surviving liver cell to proliferate. So this is our mitogen, right? There is mitogen, and the last one is the growth factors. Right, so the last one, growth factors. So the growth of an organism on organ depends on a cell growth as much as on the cell division. So if cell divides without growing, they would get progressively smaller, and there would be no increase in the total cell mass. So in single-celled organisms such as yeast, both cell growth and cell division requires only nutrients. In animals, by contrast, both cell growth and cell division does not depend on the cell cycle control system. Right? Indeed, many animal cells include nerve cells, and most muscle cells do most of their growing after they have terminally differentiated and permanently stopped dividing. So like most survival factors and the mitogens, most extracellular growth factors, right, binds to the cell surface receptor that activate intracellular signaling pathways, and then these pathways lead to accumulation of proteins and other macromolecules, right? So the growth factors both increase the rate of synthesis of these molecules and decrease the rate of degradation. So some extracellular signal proteins, including this PDGF, right, can act as both growth factor and mitogens at the same time, stimulating both cell growth and progression through the cell cycle. Right, so the compared to cell division, there has been surprisingly little study how the cell size is controlled in animals. As a result, that's still mystery. Right, so this was a positive, right? The positive signals. So the extracellular signal proteins, that the survival factors, mitogens or growth factors, act positively to increase the size of organs and organisms. But there are some extracellular signals, right? However, they act to oppose these positive regulators and thereby inhibit the growth, right? For example, myostatin, right? There is myostatin, right? This is a secreted signal protein that normally inhibits the growth and proliferation of a precursor myoblast, for example, that fuses to form a skeletal muscle cells during mammalian development. So when gene that encodes myostatin is deleted in a mice, their muscles grow to be several times larger than normal because both the number and the size of the muscle cell is increased because there is no myostatin present to stop that. Right? Cancers are similarly the product of mutations that set cells free from the normal social controls operating on the cell's survival, growth, and proliferation. Because cancer cells are generally less dependent than normal cells on the signal from other cells, they can out-survive, outgrow, and out-divide their normal neighbors, producing tumors that can kill their host. Alright, guys, so that was the cell growth, cell division, and the cell size. Right. So I think that's done for this topic. Alright, guys, thank you guys for watching, and we'll see you in the next video with the